Hello everyone and welcome to today's review session. So today we're going to be going over how to draw a membrane as well as what a membrane is composed of. So at the most basic level here, I've drawn out a phospholipid. A phospholipid is the most fundamental component of a membrane. This phospholipid is composed of two major portions. This is going to be the glycerol plus the phosphate. And again, just a reminder that phosphate's formula is written as PO43-. So that's what we're talking about when we're talking about a phosphate. And then we also have two fatty acid tails. So the primary component of how this makes a membrane is actually the affinity for the certain parts of this glycerol molecule in relation to water and fats. So the way that we think about this would be, what are they attracted to? Right? So when we think of the glycerol and the phosphate, we consider these to be hydrophilic. This means that it likes water and tends to stay away from fats. Now when we think of the fatty acid tails, fatty being in the name, we can probably suppose that these are going to be hydrophobic. So hydrophobic basically means that it likes fats, but tries to stay away from water. You can also think of it in a slightly different way, right? So philic essentially means polar. And you've talked about polarity a little bit when talking about chemicals. So with this polarity difference, the reason why polar things are hydrophilic is because water itself is polar. Now when we look at the phobic fatty acid tails, these are actually going to be nonpolar. So this helps them to be attracted to fats while staying away from polar water. So the name that we use to give all of this is basically an amphipathic molecule. What this means is that the phospholipid actually has a dual nature. So the head is completely different. It exhibits different properties than the tails do. So we consider this amphipathic. Let's look again at what the phospholipid bilayer basically looks like when we see it in a typical membrane. So, as we've mentioned, the function of a membrane is a phospholipid bilayer that separates two aqueous mediums. These two aqueous mediums are going to be the ICF, or intercellular fluid, and the ECF, or extracellular fluid. Basically, this just means that this is the fluid inside the cell, and this is the fluid outside of the cell. This can also be taken when considering organelles. ECF can also be outside of the organelles, but inside the cell, and the ICF can just be the fluid inside of the organelles. So the way that we're going to look at this is basically that we want a like to be with like, and opposites to be opposite one another. So it would be kind of strange to try and lay out a molecule this way. Because as we just discussed, these are hydrophobic, the fatty acid tails, whereas the glycerol and the phosphate are hydrophilic. So they're not going to want to be put next to each other because they have completely different properties. They're amphipathic molecules, as we also mentioned. So what this means is that the like has to associate with like. So we typically lay out membranes like this. As we mentioned just a moment ago, this is a phospholipid bilayer. So bi meaning two. There are two layers of phospholipids. And as we just talked about, they're going to orient this way. So the reason that it's the fatty acid tails next to one another and not the heads of the molecules is for one simple reason. So as we discussed just a moment ago, these heads are going to be hydrophilic. And the extracellular fluid is going to be right outside of this phospholipid bilayer. So the extracellular fluid is going to be majorly hydrophilic, right? So fluid is primarily made up of water as well as other nutrients and everything. So this is going to need to be hydrophilic as well. And as we just said, like likes to be with like. So in order for this membrane to associate with outside water without being completely broken up, we need to have the hydrophilic part facing toward where the majority of water is going to be. The fatty acid tails, on the other hand, which are hydrophobic, are going to want to associate and almost hide in the middle of this membrane. So they're going to associate with one another and basically orient this way. So now let's take a look at what the membrane looks like in total. So what we're going to basically take is just a section of the membrane. Usually this would extend around whatever it's protecting in almost a circular or irregular shape, but it's going to be a complete kind of loop. But right now we're just looking at a section of what a membrane would look like. So when we consider this membrane, this particular model is called the fluid mosaic model. So this again is still a bilayer, but the mosaic denotes that this is a membrane containing many different proteins. As we can see here, we do have multiple different proteins, which we'll go over in just a minute. Next, the fluid denotes that the protein location is dynamic and ever-changing. So if you were to look at this in a non-2D setting or an animated setting, basically these proteins would be just kind of moving throughout this bilayer. The phospholipid heads kind of get pushed aside and the proteins move because it's fluid, because it's moving within the bilayer. So now, to go over the specific parts of this fluid mosaic model, first we're going to start off with the IP. This is going to be the IP. This is going to be an IP. And this is going to be an IP. So basically what an IP is an intrinsic protein. This is a membrane protein that extends into or passes through the membrane. So intrinsic means inside. So this protein is going to have to pass through the membrane, whether it's just going through half of it, going through the whole thing, or it's going through a part of it. These proteins are used for transport, motors, receptors, enzymes, cell adhesion molecules, all those kinds of functions. So these are going to help with things coming into the cell, going out of the cell, helping things move, all that sort of thing. Next, we're going to move to the EPs, which is going to be here and here. EPs are extrinsic proteins, extrinsic meaning outside, kind of like extracellular fluid. So these can be either on the outside or the inside. And these are membrane proteins that lie on either surface of the membrane, but must touch the membrane. So these have the same functions as the intrinsic proteins. However, their location is just different. So they can be used for transport, motors, receptors, enzymes, cell adhesion molecules as well. Next, we're going to talk about glycoproteins. So a glycoprotein is going to be this guy right here. And a glycoprotein is a membrane protein, which can be either intrinsic or extrinsic. As you can see here, we drew it as an intrinsic protein. And this is going to have carbohydrates, which are organic, attached to it. So these guys right here sticking off are going to be CHO, which means carbohydrate. So these always extend into the extrinsic or extracellular fluid, so the ECF. These are never going to point in toward the cell. And these are used for self-recognition, specifically with the carbohydrates. So, for example, in the immune system, there needs to be a lot of recognition going on. So this is the primary source of that recognition, is going to be this glycoprotein with its extending carbohydrates. And then finally, just going back to a glycolipid, which is actually going to be this one, we're just going to add kind of a little hat to it. And this hat is also going to be composed of carbohydrates. And a glycolipid is going to be a lipid molecule with carbohydrates attached to it. So these also always extend into the extrinsic cellular fluid and have the same functions as glycoproteins. So to go back to that, they can be used primarily for self-recognition. So when you look at this model as a whole, you can see that it has a whole lot of functions and a whole lot of implied functions as well. When you go back to this model, you can see how things would have an easier time completing active or passive transport across the membrane, how you can recognize different cells within certain systems within your body. And ultimately, this will give rise to a lot of functions that we're going to be seeing throughout the rest of anatomy and physiology. So this membrane component is not only going to be important for your exam, but it's going to be important for the rest of the class as well, and should help you when understanding future biological processes in this class and beyond.